Let's get started. Okay, so let's get started. It's my great pleasure to introduce our seminar speaker today, and Jill Escher. I think many of you knew Jill, and so Jill did her undergraduate at Stanford, and then after that, she got her law degree from UC Berkeley, and then she practiced law as a lawyer, and for a couple of years. And then later on, she had a family. She had two children, and it turned out that her two children are with the autistic spectrum disorders. And I think that events turned her into a advocate for autism research. And so she's going to talk a little bit about her history. And so, but Jill is a well-known autism research philanthropist, and she set up the Escher Funds for Autism Research. And so she's also one of the strongest advocates for autism research. And she has been in the leadership of the in the Bay Area and in the national level, the Autism Society. And she is very, very knowledgeable. I think many of us are so impressed with her knowledge. And sometimes actually I personally go to her and asking her about, some of the information on autism research. I learned a whole lot from her. And many of my colleagues and, we all really respect her, and she's just fantastic. And so I'm so honored to have Jill here. And so she's going to talk about her favorite topic, and autism. And so, Jill, the floor is yours.
Thank you so much, Dr. Yan. I'm going to go ahead and share my screen just to get started. And go to play from start. There you go. Can you see it? Yes. Okay.
Yes.
Okay. Well, thanks so much for that very kind introduction. I'm delighted to be here down at UCLA, and I grew up in LA, and it's always a pleasure to be here, but it's even better to be talking about my real favorite subject, which is germ cells and germ cell exposures. So this is sort of my content warning slide. As Dr. Yan said, I'm not a scientist at all. I have a background. I was a lawyer. I have a degree in English. I don't have a degree in even an undergraduate degree in anything scientific. But I come to this very much from my grounding in the autism community, where I've been very, very involved for more than 2 decades. I'm currently president of a national organization called National Council on Severe Autism. I'm past president of Autism Society San Francisco Bay Area, where I remain very active. My husband and I have a philanthropic fund, and we fund not only research, but we fund a whole bunch of programs primarily in the San Francisco Bay Area. And we're also housing providers, low-income housing providers to a great many adults with autism and related developmental disabilities in the Bay Area. And then most importantly, 2 of my children have nonverbal autism. And this is beautiful Sophie, who is just about to turn 17. And this is handsome Johnny, who is 24. So when we think about the scientific method, even in this sort of high school biology kind of presentation of it, where I sit in the scientific method is firmly on the right side. What I do is not on the left side. I'm a question asker. I'm not a question answerer. But I feel like, especially in the realm of autism, we need more people who are bringing up new hypotheses, who are asking difficult questions and new questions, even if they are uncomfortable. And I am very glad to fill that role. I wish there was more of that actually in the autism world. But what I'm going to do before I really start my presentation is try to put you a little bit into my shoes as an observer and a questioner in the autism world. And we'll do that by visiting with just 8 young men who have autism. And this is just a tiny sample, right, of people I've interacted with who have led me to ask certain questions about the origins of autism, which we will get to after we talk about autism. So first, I'm going to start off with and I don't know if the sound is going to work or not. I should tell you, there are two quick videos in this. There might not be sound, but we'll try. So I'm going to introduce you to the brothers, Ian and Harry. They aren't twins. They live here in LA with their very wonderful mom. I happen to have a video of them because a new documentary just came out about them this year. So this is a clip from the trailer. I don't know if the sound is working or not.
And 9 hours a day. Keep them calm. I think my whole life was a preparation to do what I'm doing right now with the boys. He's always moving. So he, right, buddy? Yeah, my hamster. Yeah, I know. I'm just trying to keep my kids safe. It gets really simple. There we go. Here we go. Let's go. Come on. Parents like me, when we're gone, our kids will be institutionalized.
Okay. So that was Ian and Ian and Harry. I don't know how. Did you hear the sound, Dr. Yam?
For the next video, do you think you could?
Yes, a little bit.
A little bit.
Could you hit share computer audio?
Oh, you know what? It's such a short video. It's probably not worth it
Okay.
to do that. So hold on. Let me go to the next. There. Here's two other brothers named Andy and Matthew, and they live up near me in San Francisco, and they both have idiopathic autism that's not as severe as Ian and Harry, who you just saw, but they still require very, very substantial support. And let's meet some more people. Let's meet Pablito, who lives near me in Santa Cruz. His mom is actually on this call. I saw her. So here's Pablito as he's having breakfast one morning. And there you also see Luke, who has many features that are very similar, probably don't. Luke grew up about a block away from me, and there he is with his mom, Deb. And he has many features that are similar to Luke, although not as self-injurious as we just saw, as Pablito. Sorry. Oop. And here are two other neighbors of mine. These guys live within a couple blocks of me. This is my friend Ryan, who's with my daughter Sophie right there, and then my friend Harrison. And they, both have autism. They both require, very substantial daily care and supervision. And all of these cases, I just showed you 8 young men. I could have showed you dozens and dozens and dozens, but of course we don't have time. All these cases are considered idiopathic autism, but traits like theirs do not occur up their family trees, or, the siblings don't have kids, like these young men. But in speaking to their parents, and again, many others I'm not showing you, I noticed some very, very striking patterns that absolutely jumped out at me, grabbed me by the neck. And that's why I do what I do, because I see these things that unfortunately haven't been really discussed before. So before I talk about what I saw, I want to go into autism, because I know that I'm talking to mostly reproductive biologists today. And reproductive biologists may not know a lot about autism. So just very briefly, autism is a very strongly heritable neurodevelopmental disorder. Its recurrence among siblings, and we see this in my own family, of course, is about 16 to 20%. The heritability estimates are up to 80%, but strangely, or maybe not so strangely, autism does not descend down from the family tree. The reproduction rate in autism is quite low, and so it's surprising that we see this upward trajectory in terms of prevalence. And we also see a very strong male to female ratio. And this is probably the most repeated finding in all of autism research history. It's approximately 4 to 1, but it might be a little higher, a little lower. Autism is also rooted in early brain development, and we have to thank UCLA, among other research institutions, for really bringing this to light. It seems for the most part, there might be exceptions here and there, but for the most part, rooted in abnormalities of neurogenesis. And I'm talking in the mid-fetal period, neuronal migration, axon and dendrite formation, synaptogenesis, and overall connectivity. There's also repeated findings of reduced GABAergic inhibitory tone, resulting in an altered EI balance in ASD relevant circuits. Now, the mechanisms behind the abnormal brain development remain elusive in these idiopathic cases, but seem to relate to transcriptional dysregulation. And some of that work was done here in Southern California as well. So when I'm talking about autism, please understand that I also am alert to other phenomena that are very similar, to the autism phenomenon. And one clear one is ADHD, which is very much a parallel universe to autism. Different phenotype, but underneath the phenotype, we see similarities. It's highly prevalent. It's been growing in prevalence. It's at least 5% of the pediatric population. Like autism, early age of onset that seems related to subtle changes in neural structure and function. Like autism, we don't really know what's causing ADHD, but epigenetic processes that modulate gene expression have been proposed to be a mediator between genetic and environmental influences. So very, very similar to autism, but there are also other disorders that have been increasing among children. We can talk about debilitating anxiety. We can talk about depression. We can talk about a whole bunch of other things as well. Learning disorders. But for autism, I don't know if you saw this just recently. These are the latest CDC numbers. Autism has been increasing in prevalence for at least 35 years that we've been counting. This is a 20-year arc from the CDC monitoring network. We're now seeing 1 in 36 8-year-olds having autism, which is 4.3% of boys, and for the first time, more than 1% of girls. And if we go just here to California, where we have very robust data from our Department of Developmental Services, you can see the autism caseload growth since really the past 33 years. Now, this isn't prevalence data. I do have some prevalence data from DDS. It's very, very, very similar to the national CDC data, but lower because DDS only serves cases that are considered developmentally disabled. They don't serve everybody with autism. They only serve a subset. And you can see this incredible growth in cases. And when we look at birth year prevalence, it's a very similar trajectory. So it's not like there's a bunch of adults just coming into the system. It's mostly children. And then if we look at another way to kind of look at the autism data, this is LA Unified School District, kind of near here, and we see a 6-fold increase in cases over 20 years at a time when overall enrollment has been declining. We see the same exact type of pattern throughout our state. And again, these are people who aren't just labeled autistic. These are people who need special education because of their autism. And again, around the world, we see similar trends. This is Canada. This is Northern Ireland. I can show you dozens and dozens of these types of slides. And we've talked a lot. We've heard a lot in the media, and unfortunately from some so-called experts, that this increase in autism is attributable to better awareness or diagnostic shifts, when there's actually no significant evidence for that at all. And just to quote one of the CDC investigators, Walter Zaharodny, "The surge in autism cannot be explained by broadening of criteria, diagnostic substitution, or other rationalizations reflecting the hypothesis of better awareness." That might be true at the very, very highest end of autism, but it's not true for in these broad studies. So the $5 million question is, what has been causing these increasing rates of ASD? And the paradigm in autism research is, well, it's either genes or it's environment. And in genetics has predominated autism research for the last 15 years at least. And it has made some progress, but it's been surprisingly modest. I think everybody expected genetics to show us more than it has. Right now, probably maybe 15% of cases can be identified by genetic mutations. Maybe it's up to 20% if you restrict the population to a more severe manifestation. And the vast majority of these cases are de novo mutations. These aren't mutations, again, that are handed down through the family tree. In the genetics world, they're also looking at non-coding regions, but they're not finding a lot of functional impact there. And things like somatic mosaicism. There's some other ideas that are bubbling up, but again, they're not showing anything terribly significant. Right now, the major trend in autism research is to focus on common variation, normally harmless common variants, as somehow conspiring and conflating in some families to contribute to autism risk. And this is what is speculated to be particularly true with high functioning autism. No one really thinks that this is causing autism like in my kids, but none of this is yet proven. And so far, the results from studies have been very, very modest. So the other way to look at autism risk has been to look at the environment affecting the fetal development, the fetal brain. And some things, have been shown, right now, thanks to people like Dr. Rehan, right, who's on this call, more at-risk infants are surviving, and that's a great thing. But a lot of them are surviving with, substantial cognitive deficits. We've seen fetal exposure to certain drugs having an effect. Valproic acid is one of those. Maternal infection, there's been some associations between a maternal infection and fever with autism risk. There's been kind of mixed results about assisted reproductive technologies and autism risk. But again, that may implicate germline instead of the fetus itself. And there is a small contribution for sure from advanced parental age, mostly studied in fathers. We've seen a little bit in mothers as well, but that doesn't explain more than maybe a few percent of the overall increase. And again, that implicates germline. Hold on, I'm just like moving this around, sorry. But in the vast majority of cases, the causes of autism remain unknown. So my question is, can we find answers in a third paradigm? And my paradigm is, of course, because I'm talking to you reproductive biologists, what about damage to the parental germline? What about damage to the maternal eggs or the paternal sperm? And yes, I know this is not to scale. So the question is, in my mind, can some substances harm a parent's pool of germ cells that increase the risk for offspring neurodevelopmental disorders? And I think that this is really a central point for autism research, but one that, as I said, has been almost entirely overlooked. Now, but not totally entirely, because there are a few studies looking at a couple of possibilities. There was a recent study from the Deciphering Developmental Disorders study in the UK that found that chemotherapeutic agents raised the risk for hypermutation in sperm, which was related to the risk for having offspring with developmental disorders. Diethylstilbestrol, this was a study that our fund funded. There was a sharply increase for ADHD in grandchildren of women who took DES in pregnancy, and that was from the Nurses' Health study. Solvents, there have been a couple studies that find an increased risk for ASD where there's a parental occupational exposure to solvents. Tobacco, this is a study that our fund funded as well. Grandmaternal smoking was associated with grandchildren's autism traits and diagnosed autism, and that's in the ALSPAC cohort out of University of Bristol in the UK. There's a more recent study from Korea finding that paternal smoking, both before and during pregnancy, contributes significantly to ASD risk. And then I just mentioned this one's not in humans, this one's in mice. When males were exposed, autism-like behavioral phenotypes persisted to the F2 and the F3 generations. And all those are very interesting. I think they all raise very, very important questions. I'm not poo-pooing them, but I don't think that they, by and large, can explain very much of autism risk or the increasing rates of autism that we see today. So I'm going to focus on an example that I'm much more concerned about than any of the others. And this is one that was not even on my radar when I started doing this advocacy. But as I said, this one jumped out at me and grabbed me by the throat and has not let go. So let's go back to the friends that we met at the beginning of the presentation. And and again, these are just a few examples. These are not, I could go on for a long time with more examples than these, but we met Harry and Ian, and we also met Andrew and Matthew from San Francisco. In both of these cases, their mothers had undergone major abdominal surgeries. In their cases, both of their moms had, or all four of their moms, I'm sorry, had burst appendices around age 12 or 14 in both cases, and underwent massive surgeries that lasted about 9 hours or so. And in the case of Ryan, who you also met, our neighbor, his mother also had to have a major abdominal surgery, not for a burst appendix, but for something relating to her ovary. She's passed away, and she only gave me a little bit of information before she passed away. I don't have more than that. But now let's look at these other gentlemen, Pablito, Luke, and Harrison. In their cases, it was their fathers who had undergone unusual surgeries. So with Pablito and Luke, both of their fathers had suffered gunshot wounds in childhood. Pablito's father had a gunshot wound to the eye. Luke's father had a gunshot wound to the leg. And they had to undergo reconstructive surgeries as a consequence of that. And with Harrison's father, Harrison's dad was born with a cleft lip and had to have childhood surgery starting when he was very, very young to reconstruct. So what has jumped out at me with these stories and so many more is there was a common denominator, which was the parental history of unusual or intensive exposure to surgery under general anesthesia, which has led me to this hypothesis that a subset, and again, I'm saying subset, I definitely don't think it's all, not even close, of autism cases are the direct result of a parent or parents' exposures to surgery under modern halogenated anesthetic gases. And I just want to be clear, I would not hypothesize that these gases and these exposures would cause germ cell effects in the vast majority of cases. I definitely don't think that's true. I think they're probably benign in the vast majority of cases. But the effects may depend on the timing, the dose, the mixtures, the length of the dose, and the genetic background of the parents or even of the child. But even if 0.5% of these surgical exposures could later result, even if you took that just an effect in 0.5% and assume that they procreated, that would result in roughly 250,000 cases a year of some form of abnormal neurodevelopment in offspring. So my argument isn't that there's a high absolute risk. I don't think there is. I think there's probably a low absolute risk, but possibly a really monumental population risk. So I want to talk a little bit about the anesthetic gases and their effects on the reproductive system. And then also next, I will go on to the animal studies looking at whether there are heritable effects or not of these gases. So this is just an overview of these gases. They came into widespread use really in the 60s with halothane, and then enflurane was introduced. Newer ones include isoflurane, desflurane, and today sevoflurane is the most commonly used of the halogenated gases, at least in the US. But of course, when we are exposing a patient to inhalational anesthesia, we're also exposing them to a whole bunch of other stuff as well, and we have to be mindful of that. They might have IV anesthetic agents such as midazolam or propofol. They might have local anesthesia, inflammation, trauma, pain medications, steroid medications, and other drugs. And these are absolutely miraculous medications. I mean, these inhalational gases have made possible modern surgery, period, end of story. I've been the beneficiary of it after I had kids, by the way, as have many of you, and has many of your children. I am in awe of the field of anesthesiology, and I really, really respect the entire field. I don't come at this as a critic of the field at all, but from just being a realist, these gases are also poisons. That's what they are. They are poisons. They're not introduced to affect a beneficial effect in the patients. They're introduced to suppress their nervous system. And these are all small lipophilic molecules that pass throughout the body, including the gonads. So when you're exposing someone through a mask, you're also exposing their germ cells and their support cells as well. It's essentially lethal at clinical doses. The only reason we survive is because we're intubated and very carefully monitored. They are well known to be neurotoxic. We see persistent neuronal impairment in young patients who have to undergo at least more than minimal surgery. And there are many, many studies, both in humans and mammal models, that show this. They induce DNA damage in both somatic cells and in sperm, it seems, although that's been not studied enough. They are epigenotoxic. They alter DNA methylation in sperm. We'll talk about that a little bit later. They are well known to be steroid hormone disruptors. So one thing that they're doing when you're under anesthesia is it's altering the way your HPA axis and the way your steroid system is working, and that might have downstream effects, of course, on the germ cells. And then finally, they are reprotoxic. They damage germ cells and fertility. And in fact, just this year, we're talking like within the last 2 months, we've seen, highly unusually, by the way, because there's so little research on this, but 2 studies, one from China and one from Brazil, both showing quite substantial negative impacts on the male reproductive system after exposure to sevoflurane. So sperm count was decreased, sperm motility was decreased, for overall fertility was decreased. And you can see in this picture, there is even morphological damage to the testes in the rats. This was from the Chinese study. So let's get to the main question here, which is, okay, we know these things are poisonous. We know that they can impact germ cells. We know they can damage at least sperm. We don't know that much about eggs. Eggs are very understudied. But is there an effect on the offspring, right? There's never been a human study on it. So have there been any animal models? And in fact, and this absolutely, I have to say, this completely boggles my mind. They saw these effects as early as 1981. So Herman Turndorf was an anesthesiology practitioner and researcher in New York. He exposed, and he had read some studies in the late 70s showing how halothane damaged sperm. So he was concerned, well, this damaging sperm, is it going to damage the offspring, right? Smart guy. So he exposed gestating mice to halothane, and then he bred them, and then the grandchildren, so that the generation born of the exposed oocytes were shown to be significantly slower than control mice throughout their training on a maze test. And the bottom line that the researchers suggested was that the impaired learning in the offspring of the exposed oocytes suggests that the anesthetic agent halothane may have caused a genetic aberration in the exposed eggs. Well, that's quite something. And then they repeated the experiment using enflurane instead. And instead of gestating dams, they looked at sires. So they looked only at males, sorry. And again, they reported impaired learning in the generation born of those exposed germ cells, male germ cells in this case. Bottom line, and I quote, "It seems likely that spermatogenic changes caused by enflurane are associated with genetic alterations that altered the pup's brain development," right? So of course they didn't look for the genes at the time. All they saw was the behaviors, but they raised the question, and they didn't know about epigenetics at the time. What was happening in the egg and sperm. But after these 2 studies, and yes, they were small studies, but they should have raised incredibly serious questions. But guess what happened? For 34 years, there was nothing. Bupkis in the scientific literature, nobody followed up with these. And I think it's because these gases weren't thought of as mutagens, right? And if it's not a mutagen, right, well, why worry, right? Well, as we know now, it doesn't necessarily have to be a mutagen to be of concern. So let's fast forward now to the University of Florida. And this lab, Dr. Anatoly Martinyuk is a very well-known anesthesia researcher. And he took an interest in the heritable effects of sevoflurane. And he's published now 4 studies, I'll go over them very, very quickly, that have shown heritable effects of sevoflurane in a rat model. First, he exposed neonate rats. And I won't go, I'm mindful of time. I won't go over too much detail. But they did find that an increase in DNA methylation in the promoter region of the KCC2 gene, which is involved in many neurological diseases, including autism, in the sperm of the FO males, but also this increase in the hypothalamus and hippocampus of the offspring, the F1 males. And they noticed that the changes were in the males and not in the females. Bottom line, sevoflurane could induce non-genetic effect in early stage germ cells, causing some sex-specific brain and behavioral abnormalities in the next generation, even at low concentrations. He had to use subclinical concentrations of sevoflurane, and even then he had this effect. Then he looked at a different life stage. He exposed young adult rats to sevoflurane, and he saw similar results, although they were a little more attenuated, not as severe, not as notable as in the first study. And again, he saw that effect in the F1 male offspring of the exposed parents. Bottom line, the germ cells may be sensitive to sevoflurane throughout their lifespan, not necessarily just in early life. And then they did a slightly more involved study, and they used a methyltransferase inhibitor, decitabine, to see if it can kind of normalize the behaviors. And it did. But the animals who received the sevoflurane but not the decitabine, they saw that the expression of these genes that you can see here increased by more than 40% compared to the unexposed control males. Bottom line, they found that sevoflurane alters DNA methyltransferase activity and gene expression. Notably, Dr. Martinyuk was very insistent that he found that the germ cells seem to be more sensitive to the deleterious effects of sevoflurane than the neuronal cells, that the male offspring could be affected even when the general anesthetic exposure was insufficient to induce significant abnormalities in the exposed parents. Finally, they just published a 4th study, and this time they tried to kind of do something that mimicked real life, and they inflicted traumatic brain injury in young adult male rats, and then put them under sevoflurane. And the offspring of those poor rats exhibited acute and persistent dysregulation of the HPA axis, inflammatory markers, and behavioral deficits. Again, only the F1 males were affected. Bottom line, young adult males with TBI may be at an increased risk for having male offspring with neurobehavioral impairment. Again, not a shock given what we saw before. And then just last month, I think this lab, kind of after doing these experiments, said, oh my God, we've had enough. We've seen enough. We have to give this phenomenon a name. And they wrote a review, and they called it intergenerational perioperative neurocognitive disorder. Now, I probably would have gone for a more zingy name than that, but I totally understand why they did this. They've seen this phenomenon over and over again. They see that it has implications for human health, and they wanted to slap a label on it. Now, there are other labs that have looked at this phenomenon. Vesna Todorovic, who's a very prominent anesthesia researcher and a true pioneer in first noticing and describing the neurocognitive dysfunction in young children who had undergone anesthesia, had done a study on neonatal female rats, exposing them to sevoflurane. And she bred them and examined the offspring brains, but not the germ cells or behavior. So it was a little more limited. But she did notice that the offspring's brains exhibited epigenetic abnormalities, and that it was sexually dimorphic, only occurring in males. The upregulation was in males. So the bottom line was the sevoflurane exposure to oocytes can result in altered brain development in male offspring. Again, this pattern that follows what Dr. Martinyuk's lab had seen. Finally, we have a paper from the lab of Dr. Corses, who's on this call today, and hopefully will correct me if I misspeak. But his study exposed pregnant mice to sevoflurane, and he looked not only intergenerationally, but also transgenerationally. So he looked at the F2 and the F3. So the F3 come from a generation that had been completely unexposed to anything. The germ cells weren't even exposed, but the F2 were germ cell exposed. They found in the F1, which is the proximally exposed generation, that there were adverse behavioral deficits, including sociability deficits and increased anxiety. Then in the offspring, and again, the offspring of the offspring, more than 40% showed the same behavioral problems as the F1. They only tested males. And this inter and transgenerational inheritance was confirmed through ATAC-seq experiments in the sperm of the F1 and F2 generations, which showed a dramatic recruitment of transcription factors to enhancer sequences of genes that are associated with ASD, including these three, ARID1B, NTRK2, and STMN2. Bottom line was that sevoflurane induced alterations of the transcriptional landscape in the germline, changes in the progeny's neural germ cell epigenomes, and the development of behavioral phenotypes similar to those displayed by humans with autism. So I've just shared with you the research that we've seen in mammalian models. The big question is, does GA exert heritable impacts in humans, as I hypothesize that it does? And this, by the way, this is an image from a paper that I wrote with Dr. Yan and with Dr. Corses and with two other researchers, published in the Journal of Autism and Developmental Disorders in 2021. Please see that for a lot more detail than I'm giving you today. And the answer is that there are almost no human studies on this question whatsoever. So like, you know, again, it boggles my mind that we really haven't even begun to touch this question. But there are a few things worth mentioning. One is this study from Denmark, 2016, Roman Barres group. They looked at the DNA methylation in human sperm after men had had bariatric surgery. And they found significant changes in sperm DNA methylation in 1,509 genes, approximately 1 week after surgery, with persistent effects in 1,000 genes and more than 1,000 CPG positions a year later. Now, Dr. Barres, I actually talked to him personally about this. He attributes the changes to nutritional changes. Dr. Martinyuk certainly, and I certainly think it's probably more likely to be attributed to the surgical exposure. There was also a study from Yale a couple of years ago that, again, from Denmark, looking to see if autism was associated with preterm birth, and they found a very strong association, a twofold higher risk for ASD, where a parent had been born very preterm. Now, this was not an anesthesia study, obviously, but as probably Dr. Rehan can tell you, if you're born very preterm, you are at a sharply higher risk for having early life surgery. For example, heart surgery or hernia surgery. So that wasn't part of the study, but it's a little bit of speculation I'm throwing in. Finally, there's a study that we funded, and it looked at old anesthetics to see if maybe they were exerting a heritable effect. And this was in the collaborative perinatal cohort, which is very, very, very old cohort, but the data is still out there. And I thought, well, there's no way that the old anesthesia is having an effect, because we really didn't see a spike in autism, back, in my generation. But they actually did find some subtle effects. I won't go into it here, but if you want to look it up, you can see that old anesthesia may be having some effects, but nothing really to write home about. So, given that there's so little research out there, this is really my cry, that germline exposure to modern general anesthesia should be a priority for autism research and for reproductive toxicology. And, it's a shame that after those NYU experiments in the early 80s, almost nothing happened. I mean, we have 3 decades of catch up to do, in my opinion. So why should it be a priority? First of all, it's biologically plausible. We see it in these repeated rodent studies. We also see that the dysregulation of early neurodevelopment that we see in autism very closely tracks the mechanisms at work with GA, right? So there's a very strong parallel between the effects of GA on a molecular level and how autism arises in early neurodevelopment. So I would guess that those genomic pathways overlap. And really, this is what's quite striking about this hypothesis, is that it would actually, unlike really any other hypothesis I've ever come across, it would help explain the major mysteries of autism. It would help explain the dramatic increase in autism prevalence and the timing of this increase. It would explain the high recurrence rate among siblings, because if we're talking about affecting the pool of germ cells in a parent, right, the spermatogonial stem cells in the male, or the ovary, the oocyte reserve in the female, you would expect there to be a high heritability, right? You would expect a high recurrence rate among siblings. It would also explain the high male-female prevalence, which is something that remains very, very poorly understood. But what we saw in these rodent studies was that the males were far more likely to be affected. It would also help explain the heterogeneity of phenotypes, because of course, the exposures are all different. You have different mixtures, you have different timing windows, you have different underlying genetics, you have the doses are all different. There's so many reasons that that you might have heterogeneous results from exposures like this. Now, the mantra, including here at UCLA, the mantra in autism research has been genetics first, genetics first. It's been genetics first for a long time, and we're pouring literally billions of dollars into autism genetics. I would argue that it's time to put genetics first to bed and to move to a genetic toxicology or epigenetic toxicology first mentality in autism research. It's here where I will argue we will find fertile ground for actually figuring out what's driving some of this increase. So most of you on this call are researchers, and I hope that I've kind of raised some questions that maybe deserve some follow-up. And so the last slide here is about, well, what are the next steps? Well, I feel like I've done my work on the right-hand side. I've been an observer. I've been an absolute nagging, annoying questioner, right? Not just of researchers, but of people throughout the autism community and families. I've been a hypothesis driver. But the next steps are up to the research community and definitely not up to me. And there are a lot of things that can be done. I mean, first of all, very simply, we need to do studies on GA-exposed human egg and sperm, period. That has to be done. It's shocking that so little has been done. We have to look at the sperm of autism fathers, but in light of their exposure histories. There are a few studies that look at the sperm of autism fathers, interestingly enough, and all 3 have found abnormal DNA methylation in their sperm. But of course, that they didn't relate that to any exposure history. They were just looking at the sperm, kind of in a vacuum. There's also a study out of UCSD that finds high sperm mosaicism in autism fathers, which is interesting. And then, of course, we need all of the animal studies that can actually look at the behaviors across generations and the mechanistic studies. And then in autism research, we need to do more intergenerational epidemiology. Almost all the epidemiology has been looking at, proximal exposures to the fetus and not looking at the parents' histories. Autism genetic studies are still highly funded, they're highly preferred, but they are a very important resource if we can incorporate this exposure paradigm to help explain heritability. I think it would really help these genetic studies find more answers. So I'm often on the tails of these researchers, asking them to look at these questions. So far, I have not, sadly, I've not made a lot of progress with them. And also, we just need a critical reevaluation of how we consider heritability and sibling recurrence. It's always been assumed that that's genetic. And my argument is that's not necessarily genetic. Finally, I don't have a lab to thank. Obviously, first and foremost, I want to thank so many autism families across the country who have shared their stories with me, who have put up with me emailing them and badgering them with questions, and who have given me permission as here to share their stories with the public. And I'd also like to thank so many researchers, much more than I've listed here, but researchers who have helped me and inspired me along the way. And there's my contact information. And that's it, Dr. Yan. I think I kept to my time. I just checked.
Absolutely, you are doing great. Thank you so much, Jill, for the wonderful talk, and it's very inspiring. And as usual, I learned a lot. So now we're open for questions. So if you have questions, just unmute yourself and then start talking. Or maybe you can leave your comments or your question through the chat box, and I will read it to Jill. So go ahead. Victor, it seems that you have questions. Somehow I cannot hear you, Victor. Sorry. Okay. Gonna unplug your earphone or whatever. But still cannot hear you, Victor.
Maybe type in chat.
Yeah, so. Okay, now we can hear you. Go ahead, Victor. Okay.
It's hide and seek. Hide and
Te
seek with Victor.
technical problem there.
Yeah.
You know, other people. And so yeah, go ahead.
Well, thank you so much for the talk. That was so many things I didn't know, and I thought that I knew some things about autism. So thank you for that. You know, I'm 23. I really hope to have kids at some point, and it's kind of scary to hear a lot of that in your talk, because at least at my age, people are getting their wisdom teeth removed. You have to most likely go under, anesthesia. So I guess my question would be, as someone who ideally would like to, avoid having a kid that struggles a lot, not that it's inherently bad to have autism, but if I can't avoid it, should I try to avoid the anesthesia like you mentioned?
Well, honestly, I think it's premature. As I said, I'm a hypothesis raiser. I'm not a hypothesis answerer. And obviously, if someone has to have surgery, I would never say to them, don't have surgery. You might have a child with a neurocognitive disorder. Like, I would never say that. You should obviously follow your doctor's advice. I mean, but that said, I think that the field of anesthesia has been completely oblivious to the potential damage to their patients' germ cells, just completely oblivious. And I think it's incumbent on the field of anesthesia, and it's incumbent on, the NIH and, the field of reproductive biology to try to figure out, like, are some things really worrying? Are some things actually safer? Like, if you're getting your wisdom teeth pulled, you're probably under propofol, which is an intravenous drug, which probably doesn't have these effects. I'm just guessing. And it's also very brief. But my point is, we need the research to figure out where the risks are, right? And then we can reduce those risks. That's all. I wish I had a good answer for you.
Thank you.
As I need to follow along, and I really think that any exposure has something to do with the duration, the dosage, and then the time window, and there's so many factors there. So I think that Jill is putting out a hypothesis. It doesn't mean that anesthesia is bad. It just depends on which one and then which specific conditions that might be associated with the autism research. As a matter of fact, Jill, I think the Escher funds for autism research has been funding a couple of projects in this context, and hopefully in the next couple of years, and we'll have a better answer to your question. So
Yeah, I hope these are, I fund pilot studies just to sort of kickstart this field, but obviously I can't, I alone can't do it. It requires NIH level, funding.
yeah, Victor.
Yes, can you hear me now?
I
Yes.
do.
Yeah. Great talk, Jill. Always very inspirational. So, I agree with you that it's really hard to do human studies. Is if you want to look at a sperm, for example, it requires somebody to donate the sperm right before the surgery, and then a few days after the surgery and stuff like that. And I've tried to convince people in the anesthesiology department here, and there's just no way. Anyway, I wonder if one could go backwards. This is what I'm thinking. So, obviously, an exposure to anesthetic gases is not causing, as you mentioned, is causing an effect only in specific cases. And one question is, as you also pointed out, is why? And there could be other exposures that person is being exposed to, other things, steroids, whatever. But the other possibility is that is the sequence in the genome. In other words, is common non-coding variants that normally don't do anything to us, so that we are perfectly fine. But those variants make us especially sensitive to certain exposures. So it's a combination of epigenetics and genetics. And so one could test this hypothesis by starting with a population of people with autism that like those 8 people that you mentioned in your talk. In other words, I think if you had, you wouldn't need a very large population, but if you had specific cases like these 8 families, and one could sequence, I mean, I guess you would need more than 8, but one could sequence the probants and the parents and try to look together with a control population in which this didn't happen, try to look for sequence variants. And it only costs, these days, it only costs, $300 to have a really good whole genome sequence. So one thing that you have that it seems to me you don't make enough use of is this knowledge and these contacts. And so, in your Society for Severe Autism, you must have a population of people with severe autism that are not participating in research, or if they are participating in research, is together with a very large population that have very weak phenotypes.
Yeah, can I interject here for a second? So I sometimes meet families with 3, 4, 5, even if you can believe this, 6 offspring with autism. I have emailed the world's leading autism researchers saying, I really think we need to do a case study on these families, right? To see both the genetic, epigenetic, exposure history, the whole 9 yards. And I don't get bites. Anyway, I think there has been like 1 or 2 case reports on some family history. But anyway, I agree with you. It's an untapped, I think, gold mine. These families, especially with multiple kids, a completely untapped gold mine.
Yeah, but I think in those studies that you mentioned, these researchers that you mentioned that are part of really of large cohorts looking at the genome, I think their idea is that bringing this family will bias the population that they have. And so they are not willing to add these populations to their studies.
Well, thank you, Victor, for the question. And Jake, I think that you have a question, right? Go ahead, Jake.
Yeah, I hope I'm unmuted. I kind of want to just about understand a little bit, I think, of just, the argument I think you're setting up. And I think it was kind of a great figure that you showed with those two new papers that just came out. I mean, they showed the exposure to the anesthetic had a huge drop in the size of the testy.
Mhm.
And I think this is more, I want to set up kind of the biology side. So are we thinking that this messes up like GnRH secretions or something that's going to cause spermiation, and that's why you're seeing this huge shrink in the testy size, because you're losing those tails, and that's what you're losing your tube plumpness, and that's why you're seeing this huge decrease? Is that, I think this is really kind of more of a Victor question, because he's the brain guy.
Well, I would read those papers, right? Because some of this stuff, again, I'm not a researcher, and so
Yeah.
some of those details are above my head when it comes to those like
Sure.
anatomical details. Like I don't know the anatomical details, and whether it's what mechanism is causing it.
Yeah.
It seems to be multiple, because you have effects on the steroid system, you have effects on oxidative stress, you have
Yeah.
direct toxicant. I mean, it's multi-level.
What I want to get at is, if it is something like GnRH, that should be something that you can measure in blood. And that's something that probably would routinely come up before potentially a surgery and then after a surgery. There might be labs that they could pull that might be samples where they could see, hey, look, we're seeing T levels drop, or we're seeing something. If you could find a biomarker. And I know that really goes to a lot of the genetic side, but I think
Jake, I'll try to address your question a little bit.
Yeah.
So I don't believe those kind of morphology is truly representative of the whole mouse population that received that treatment. The bottom line is this: even though you have a GnRH abnormalities, you're going to see these things in both tests. The fact is that one test seems to be normal, the other test is really small, and then those really occasionally we see that maybe like due to some damage or some just like it's a probably it's outlier. It's not. I mean, if that happened really to that anesthetic treatment, that's toxicology. It's the basic kill all the sperm. I really think that this exposure to general anesthesia, the direct impact on testis morphology will be really minimum. And then I think that the most things happen behind the scenes. So basically, it's undetectable. It's not discernible through gross morphology. That's my take on it. But anyway,
do you think it might be non-coding RNAs?
Of course.
Well, I think
there she is. This is the star of our show. Lisa Ju. Hi, Lisa.
I just want to share the results in our lab. Actually, we do the neonatal anesthesia and then collect the testes. We also find the similar phenomena that maybe in 10 rats, only 1 or 2 rats have the smaller size of testes. It's true. We also found this, but we didn't publish it because we cannot believe it. And we do the morphology. We do immunohistochemistry, and we do slice, but we didn't see the structure change, no morphology change.
So they still have spermatogoa. They're not losing the late.
It's very apparently. Maybe he had 2 testes. Maybe we get one is normal size, another one is smaller, and we do morphology, but no change in it. So we didn't continue doing.
I mean, I think.
But it's true exists. We have no idea how can it happen.
If there was a way to tie this to potentially having a small cohort study on those families to be able to show that autism children have a change in the chromatin landscape, and then to tie that back to some exposure, that would be the biggest link to the phenotype. I think that's the big smoking gun that I think it's missing.
Yes. But we try to detect the stress, the hormone, the testosterone, the estradiol. There's some change in the testosterone level.
By the way, that's Lingsha Ju from Dr. Martinyuk's lab at University of Florida.
Cool.
So she's responsible for those 4 papers that I talked about, and also that what did you call intergenerational perioperative neurocognitive disorder?
Yeah, we always focus on this one. Really interested. We find this phenomenon. Oh, cool.
Yeah, Jill, I have a question for you. And so 2 questions actually. This is the things that we're facing in terms of initiating a study on ASDs. And so one is that there are two things. One is biological samples. So do you know any resources where we can access, the large cohort of not only the patients with ASDs, but also their parents? And then so where do we have access to, those samples? And a second thing is that inevitably we have to use animal models. So I thought there should be very well characterized animal models, either in mice or rats, where you have very well defined behavior tests, like a set of, or maybe 2 sets or 3 sets of behavior tests that basically define the ASD phenotype in mice or rats. Actually, I couldn't find any. So this is 2 things I feel very frustrated. And one is that the large cohort of human samples, and the second thing is that are very good mouse models that really truly reflect the behavior of autistic humans. And so do you know any information on these 2 aspects?
Well, okay, human cohorts, it depends what you mean by samples. So there are plenty of cohorts where there are DNA samples, right, where they've collected blood or saliva, right, so that they could do usually whole exome sequencing. And the most notable one is called SPARC, and it's funded entirely by the Simons Foundation in New York City. And they already have, 40,000 participants, I think, even more than that in SPARC. It's massive, and they intend to continue growing it. But those are the only samples they collect. They do also collect some phenotype information and some family background information, but not much. There's a big study out of University of Toronto, Stephen Scherer. I don't know if any of you know him. It's sort of the successor to a cohort started by Autism Speaks many years ago, or the forerunner to Autism Speaks. And they have blood samples. And Simons also has some smaller cohorts than SPARC with blood samples as well, and they have different names. And there are smaller cohorts around. It just depends on what tissues you need, and how much information you need. Some of these have like almost no phenotype information, right? All they have is like, oh, autism, check mark, and then you don't know anything else about them. It's a little hard. And then your second question was about animal models. I am definitely not an animal model expert, but I do know it's very controversial in the field, like what constitutes, what behaviors are relevant to autism, right? From a mouse or a rat model. My argument is a little bit more pragmatic. My argument is, if you're seeing abnormal behavior, period, whether it's abnormal reproductive behavior, sexual behavior, social behavior, feeding behavior, whatever, if you see abnormal behavior, period, it should sound alarm bells. And I don't care if it's direct parallel to autism, it probably won't be, right? But it's sufficient to show that it's having this biological effect. So I don't know. I sometimes think people worry too much about the finer points.
Okay, thank you. So any more questions?
Yeah, so I have a question. Do you think that it's clear that this is a homogeneous group of individuals by diagnosis? Or do you think that it is a more diverse group of causal factors, for instance, that result in somewhat similar type of manifestations?
Yeah, that's been a big question in autism. Pretty much no one in autism really thinks that autism is a thing. I mean, pretty much now there's a consensus that autism is a variety of overlapping manifestations that arise from a variety of biological phenomenon, most of which are not understood. Now, I talked about roughly 15% of autism cases having a genetic origin, right? Those are rare syndromic cases where autism is a feature, but just a feature, right? And those are kind of their own things. Those syndromes exist in their own bubbles. And then you have probably about 80% of autism cases that are idiopathic, and we don't know what biology really is driving these features. But it's thought that it probably has a wide variety of origins. My argument is we have to look at the germline programming to understand a subset of those. And I think we will see, a great diversity the more we look into it. I mean, there is a bottom line though. And there is something that's seen really broadly across these cases, which is the early stage transcriptional dysregulation, of the mid-fetal brain. That's pretty clear. And something's causing transcriptional dysregulation that causes neurons to not migrate to the proper place in the cortex. That's seen over and over again. It's not the only thing, but UCLA, your colleagues do that work, and it's a clear trend pattern.
So are there any data on, I'm sure there must be, data on dynamic imaging of the brain in order to determine the similarity or differences in the differences that occur in,
Like a functional MRI, like fMRI kind of imaging, or
there are a number of techniques that can be used that are
yeah.
in
Yeah,
the-
I'm not, yes. I know it up at Stanford where I live, they've done work on that. UC Davis has done work on that. I believe UCLA has as well. And yeah, they do see a number of functional differences in terms of how the brain connects, during various tests. But I'm not an expert on that. But yeah, there is some literature on that for sure.
So one of the problems you have when you deal with a condition, a syndromic type of picture, where there may be multiple causal factors and multiple differences in the cause of the disease, is that in order to study things like epigenomics, you really need to be able to narrow your study group to have the similarities. This gets to the issue that Wei Yan raised earlier about having a mouse model where you have a population of rodents that is impossible to duplicate exactly in humans, that will allow you to be able to look at a group that has the underlying genetic background that would also allow you to look at epigenomic phenomenon in a more easy fashion. Is that fair, Wei?
I'm sure. I don't know. Well, my only comment on that is, I would just like to see basic toxicology done. I mean, I agree with, I see what you're saying, but about, getting these homogeneous groups, we still just need basic toxicology on this exposure and on the reproductive system and heritable effects, and that's where I would start.
All right, I see, Chi, do you have a question? No? So you have to unmute yourself, you're fine, right? And I see Dr. Peter Schlegel is here, and Peter, do you have a question? No?
So I found this fascinating. Thanks so much, Jill, for bringing this all together. I wonder if you really have a link between individual agents and this rise in autism that you're seeing. And just as a footnote, I'd be careful about, saying that things that are non-inhalational agents like propofol are safe. I mean, propofol has, to my understanding, the greatest volume of distribution in the body, and it particularly loves lipids.
Okay, well, that shows you my, as I said, I am a naive non-scientist. I think I said it's probably less damaging because I looked at some literature looking at DNA damage caused by inhalational, the volatile inhalational anesthetics compared to propofol, and propofol was less damaging to the DNA than were the halogenated anesthetics. And so I think that sort of stayed in my mind as maybe, it's overall less damaging to the genetic, molecular milieu. But yeah, I mean, you're probably, none of these things are non-poisonous. They're all poisonous. They're all lipophilic. And you're right.
Yeah, and Dr. Schlegel, I just want to update you a little bit. Actually, Jill gave out a grant to a junior investigator whose name is Matthew Leon, and I'm working with him to test the combination of a sevoflurane and a propofol, and it's a combination of different dosage, different exposure duration.
That's right.
And of course, we're using mice, and so we're halfway through this entire study. I think by the end of August, we should have all the data, and so I will update you if the data look promising. Regardless, I'm going to update you on that.
Thanks.
Thank you. So if no more questions, then we'll have to stop here. And let's thank Jill one more time and for the excellent talk. And so Jill actually is physically on campus. And so I'm not going to keep the center people here anymore. And so later on, we're going to have individual meetings. And so we just stop here. Thank you again for all of you for participating in this seminar. And so we're going to have our next talk from McGill University. I'm going to update you on the seminar information 2 weeks from now. Well, thank you so much. You guys all have a good day. Bye.
Thank you, everyone. Thank you, Dr. Yan.